in every human cell, there are RNAs that are being transcribed in the nucleus of cells, and they eventually get exported into the cytoplasm. So we care about RNAs, like you guys, non-coding RNAs, RNAs that are made in the nucleus that eventually get processed. And so in these processing steps, these are all mediated by both epigenetics, of course, and also proteins that eventually bind directly to these RNAs. And so these are RNA binding proteins that we care a lot about. And in recent years, more and more of these RNA processing steps are being implicated in human genetic diseases. And they may arise because of sort of defects in the binding sites of these RNA binding proteins that interact with these RNAs, or in the RNA binding proteins themselves, right? And so our work is set up to identify where these RNA binding proteins bind to non-coding RNAs as well as coding RNAs, and how they mediate processing or proper processing of these RNAs in human cells. And we are trying to establish the map of all these different RNA binding sites in the human genome, and eventually hopefully build these sites together to generate better predictive models of human disease. And in the brain, there are a lot of these RNA binding proteins that are specified and expressed in a cell type specific manner. So I believe a report by Pam Silver's lab has shown that about 2/3 of RNA binding proteins out of, I believe, 300 RNA binding proteins are expressed in a very cell type specific manner in the brain. And so clearly there are targets of these RNA binding proteins that have to be bound and recognized and modulated and processed in a very cell type specific way. And I'm very curious to see well, first of all, this tells us that RNA processing events, these kinds of processing events, seem to be very important to specify proper brain function. And it'd be very curious to see in the future how many of these different RNA binding proteins or events will become implicated in human disease. But I guess more today, more urgently, it would be very interesting to study how these different RBPs play parts in ensuring proper cell type specific maintenance of RNAs, and how that actually impacts neurobiology. That's a piece of the area which in some sense encapsulates different things that are happening in my lab, and so that's quite interesting for us.